Hi, just a second. Uh, yes. Okay. Uh, Hello, welcome. So this is the fifth session for problem solving for the course Animal Physiology. And today we will continue few of the topics that we discussed in the previous session. And we go into action potential in depth. If you remember, lecture 5 notes had mostly action potential. So basically, how does the nervous system works? How does those neuron they relay the information? What guides how action potential are traveling? Why are they first of all generation? So all these topics that are related to action potential and your like the propagation of this action potential so that the two neurons can interact with each other. That was the main topic of week 5, and that's what we're going to do the questions on. And like always, so these, if you remember, if you look at week 5's topics that were covered, that was mostly about the neural system. So as was discussed in the lecture sessions also, it began with origin of biological cell, because we need to first understand cells so that we understand which cells are involved in this relay of information. And that once we understood cell, which was a very brief topic that the professor covered about evolution of cell, how did the cell, the closed structure of cell originated. And then he discussed about excitability in cell, which was something that we had already sort of discussed in the previous sessions as well. So what is excitability? What do you mean by ability to get excited? What is the excited term relates to here? Then once you knew what the cells are which are excitable and which can get excited or have the potential to get excited, how do they do that? So ion transportation in the cells, so your generation of the action potential. And then next fourth lecture was about the propagation in the neuron. So you have signal generated, the signal is propagated. And the last lecture of that week was neurotransmitter and action potential, which is basically completion of the entire circuit of neuron transmission, right? Okay. So I will not go into the details of the this topic. This is just here to remind you what all you did in that section. So basically, in the lecture, what was discussed was how did you get the proper biological cell or the proper formation of the biological cell that we see in photos in eukaryotes, which is basically your plasma membrane, your cell organelles, your nuclear material. So if the main agenda of the this topic was to highlight two things. First, it just wanted you to imagine that we started with an ancestral prokaryotic cell. If you know the difference between prokaryotic and eukaryotic, you can look at the images and you can start visualizing the differences, right? So you're an ancestral prokaryote which started the form with which changed to have the pronucleus and the endomembrane system, and then over the time gradually with the incorporation of mitochondria and chloroplast. If you know, these are foreign bodies, these are foreign DNA. They were symbiotically taken into the cell. So ultimately, what you see now as a cell structure is it has a phospholipid layer and phospholipid bilayer, and you have mitochondria, plastid. These are some organelles with their own membrane, and then you have several other organelles in the cells, right? The main highlight of this part of the lecture was to make you realize that the cell is enclosed by a plasma membrane, and the importance of that will come when you do the electrical conductance in of action potential, right? And that's how you connect it. So this structure that you see here, this is phospholipid, and it is a two layer. So there on the membrane you will see there are two layers of phospholipids. So that's why I call phospholipid bilayer. And on this layer you have these different kind of gates, which are pumps, or you can also call them gated channels, and which are responsible in depolarization of the cell. Now Yeah, I'm so sorry that I got disconnected. So I was referring to depolarization. Whenever you said we had, so if you remember in that week, you had several terms related to polarization: depolarization, repolarization, hyperpolarization. So all of these terms are in relation to the iron concentration, or the flow of iron across the cells, right? So when you see any depolarization or any thing related to polarization, you are basically talking in terms of ions, and ions especially sodium ions and calcium ions. Now, this slide gives you a brief understanding of what do we mean by depolarization, or what does it mean by polarization. So depolarization is when anything is getting the word D would mean anything which is moving from its normal state. Here, normal state would mean, so you have a plasma membrane, you have certain concentration of sodium ions and potassium ions inside and outside cells. Depolarization will mean that these concentrations have now got interchanged or have now changed. So you're getting depolarized, right? And what happens exactly, you can see there is an influx of sodium from outside to inside, and you have an efflux of calcium, potassium from inside to outside. So you can see these are the two ions that move inside and outside and change the ionic concentration of your cell, and which gives these different charges across the cell, and that gives the electrical charges, which we discussed, if you remember, 70 millivolts and 90 millivolts. So we were discussing that in terms of the charges inside the cells, and because you can imagine so many calcium, potassium going outside, coming inside, sodium going outside, sodium coming inside. So this movement of charges will change the overall charge of your cell across inside and outside the plasma membrane, right? So before, let's not make it more complicated by using terminologies. Let's directly go into what exactly do we mean. So this movement of cells, ions inside and outside plasma membrane is due to action potential. Right now, action potential has 5 stages. If you remember, this is, so there are terminologies in this week or the week's lectures which are pretty easy to understand, and all throughout the lecture 5 that I had, lecture week 5 lectures, I have noticed that the topics are repeated. Mostly these are covering around action potential, how they are generated, which ions are like moving across the cell, membrane. So if you understand once, or if you just try to remember once, you pretty much sail through this week's course, right? Okay, so there are few terminologies that are important. So let's begin with the first one, which is action potential. Right. So now action potential is basically these entire chain of ions, sodium and potassium, moving outside the cell and inside the cell, which leads to a disturbance in the charge and the potential of the cell, and that relays an information to the via the neurons to the next cells, right? So it propagates the messages. Now, when you see an action potential moving, you need to first understand what are the steps of the action potential, or what are the phases of action potential, right? So you see action potential begins with a resting phase, which is called the resting potential. Resting potential is when you basically, nothing is moving outside and inside the cell, and it will only begin to move outside or inside, or will start showing the change only if it crosses threshold stimulation. Okay, so you keep getting this information, but when the cell decides to send the sodium out and potassium inside is when it reaches this threshold stimulus. Other time it gets initiated, but it does not lead to a action potential. It just, there is a minute change in the charges, but it does not lead to an action potential, right? So the first step is called the resting potential. Around resting potential, or around cells when they are not excited, the voltage is minus 70 millivolts, right? So this time the neurons are at rest. And why is it minus 70 millivolt? It basically means the inside of the cell is more negative, or negative compared to the outside. It means you have more of negatively charged ions inside the plasma membrane than outside the cells, right? And you, like we have seen here, and we will get to know the name in a while. So this pump here is responsible for maintaining this potential, and the pump is called the sodium potassium pump. This you need to remember because these are the exact players which take part in action potential generation. So sodium and potassium pump, easy to understand. There is another term in that pump which is called sodium potassium ATPase pump, right? So I will explain the pump when I come to the next slides, right? For now, just remember that this pump, which is there on the plasma membrane, is responsible for the 70 millivolt at the resting phase, right? Now, first step is depolarization, when suddenly there's a shoot of sodium potassium inside the cell. Now, how does that thing happen? That thing happens when these gates here open up. So these gates open. As soon as the gates open, the sodium ions keep moving inside. So your sodium ions have now started moving inside. And as your positive charges, because sodium are positively charged, they keep moving inside, the voltage or the charge of the cell also decreases, or it's rather increases. You have minus 70 to a plus 40 millivolt charge now inside the cell. And you see this sharp shoot up, right? This is called upstroke. So you see a sharp upstroke, and the cell potential, or membrane potential rather, now reaches to plus 30, plus 40 millivolts. So from a minus 70, it reaches to plus 40. Okay. Now, after it reaches plus 40, you see a sudden drop, right? Now, how do you see a sudden drop? That means, again, it has to go. So you were getting positive charges inside the cell. Your positive charges were increasing. Now, you need to go negative again. So you need to push this positive charge outside. What do you do? Which positive charge do you put outside? You put the potassium ions outside. So you have taken sodium inside, you push the potassium outside. Now, when you're putting the potassium outside, you see this downstroke. Suddenly, all the positive charges are now moving out. So you're seeing a dip in your voltage or membrane potential. And this is called repolarization, because something which is depolarized, if you are bringing it back to normalcy, it is basically you're returning to the normal state, you are repolarizing it. So your repolarization. Now, what happened here is, here at repolarization step, these channels opened up, which basically pushes these potassium ions outside. So first step was these inside, second step was these outside. So potassium ions are being moved outside. So your membrane potential again begins to drop towards a negative value, right? But it should have returned back to the normal state. This increased from here, they should have increased here, but that's not what is happening. What happens, it goes way negative than it is supposed to be. It should have come back to minus 70, but it goes very negative. And this step is called hyperpolarization. What does hyperpolarization mean? It basically means, so just remember one thing, these channels, they are not perpetually opened, because if they are perpetually open, then your calcium, potassium and sodium will keep moving, and there will always be a charge generation. That's not what happens, right? You take some ions inside and you close the pump. So sodium pumps, after pushing the sodiums out, or rather taking the sodium inside for that matter, taking the sodium inside, they will close. But as compared to this pump or this channel, the potassium channel will close slowly. Now, because the potassium channel is closing slowly, or more of your K plus exit in a given time, and because most of them are exiting, your negative charge really drops. So you really get very low millivolts, right? Potential. So you can see it goes up to somewhere around minus 90. So from a plus 40 to minus 90, just imagine the amount of charge that would have left because of the slow closure of your potassium channel, right? And once that channel closes, again the voltage comes back to resting state, which is basically minus 70. So you have these entire 5 steps starting from resting phase. Then you have a stimulus, takes it to depolarization, repolarization, goes way below hyperpolarization, and then you have a resting phase. This you need to like understand better, and you need to remember terms and what happens in those exact stages, and that's what will define the action potential to you, right? Okay. Now, related to action potential, there was a very important topic that was discussed in the lecture, which is Hodgkin-Huxley model, if you remember. I will not go into too much detail, just like how professor did not go into way too much detail, but basically why they are on the same slide. This model explains this potential, how propagation of action potential takes place. All these complicated maths that you see, they're basically trying to give you an idea that how this action potential they travel inside the cell, right? Now, what all terms do you see here? So if you see this, you will remember something like circuit, and this is what he was discussing in the class also, in the lectures also. So circuits. So this model basically tells that the membrane, which we see here, it basically is a electrical circuit. Okay, and in the electrical circuit, which I think will be here. Okay, so it's there also, but let me just explain from here. In the electrical circuit, you will have capacitor, you will have a resistor, and you will have a battery. All these are also present in the membrane in different forms. For example, capacitor, which is basically storing the charge, it's in the lipid bilayer because they are the one that open and close the channel to move the ions inside and outside. Then you have resistors, which are responsible for the ion conductance, and you have ion channels, which are basically the channel by which your Na plus move. And then you have battery, which is basically the ones that maintain the charge. Those are the ions. So you have ions, you have the channels, and then together you have the capacitor, which is the lipid bilayer. So they all function in a way that your electrical circuit functions, right? So basically you have your voltage gated Na plus channel. These will open when they are depolarized so that there is Na influx, and then they will get inactivated, so they will close. Then as soon as these concentration increase, this channel will increase, open up voltage gated K plus channel, and the K plus will move out. They are also leaky channels because they are there are some channels which remain open, for example, these kind of channels. So you also have background currents, but mostly you have these three players. You have ions, which are acting like battery. You have these channels, which act like conductors or resistors. And then you have these capacitors. So all these make up or act like a circuit. And this resemblance to an electrical circuit is what was meant by the Hodgkin-Huxley model. Why is it important? Why are we even talking about this like this? Because it is the first quantitative model of action potential. And why quantitative? Because it is not just showing you it goes like this and like that out. They basically gave the like the mathematical basis of these propagation. So that's why it's the first model which had quantitatively shown the action potential. And he showed how these ion channel kinetics is basically how these ions are moving across and what is the mechanism and what is the rate of these movement. So the ion channel kinetics was also shown by this model. So you see, before biologists would only consider this as a movement outside and inside, and that would just rest at that. But when you give a mathematical basis to it, you basically have a very strong position to explain the method. And that's why things like biophysics or structural biology, these make more use of mathematics to explain the biological phenomena. And this is just one of those examples, right? Now, you have gated channel, like I just mentioned. Your gated channels will behave like capacitor. You have ion channels which behave like conductance. You have ion pumps, which are basically these ions, they behave like batteries. So basically all of these are part of the circuitry of the action potential, right? Now, one thing that you need to remember, which I had kept uh, like I didn't tell you yet, was number of ions that are moved out and in. Okay, so remember the number of ions, which I am not discussing here, so that because I have questions on them, then I will discuss them, right? And this is just what I was saying. So this is the plasma membrane, and this is how it is equivalent to the circuit. So you have the capacitor, you have the resistor. This is how you draw a circuit, and this is how you can draw the analogy between the circuit and the plasma membrane. Okay. Okay, and briefly also topics that were touched upon were central nervous system and the cells of CNS, which is their part of the questions I will show. And the last thing that was again discussed was the, if you remember, synaptic transmission of information. So let me remind you, basically you have these neurons, and you have the myelin sheath, and you have something called nodes of Ranvier. Let me give you all the terms, and then in my questions, all of these will come together for helping you understand the topic. So you had nodes of Ranvier, you had myelin sheath, you have conduction like saltatory and so basically saltatory conduction. And then when it reaches the end of the neuron, there is something called exocytosis of neurotransmitters, which is basically taking information from one neuron to the other neuron, which so basically this is action potential. Let's say this is the first neuron. So this is the action potential, and it has led to some discharge, and some new action potential has come to this end. Neurotransmitter will get released outside, and the next neuron will take this neurotransmitter into the action because of the receptors. And once the second neuron receives a neurotransmitter, it's like, okay, so I also received a neurotransmitter which basically tells me I need to get excited. So we will understand excitatory. So I need to get excited. I need to start generating an action potential. So the next neuron will now generate an action potential, and that will relay the information till its end. So you have this long chain of one neuron connected to other neurons by several dendrites, and all of these are doing the same mechanism. The information is relayed by neurotransmitter from one neuron to the other neuron, because there is a synaptic cleft between two neurons. That cleft is basically a hollow space where the neurotransmitters travel, and basically it initiates the action potential for the next neuron, right? So that's about signal transduction. And this is about action potential propagation. So you have the myelinated sheath, you have nodes of Ranviers. It's all, so if you have nodes of Ranvier, or if you have this myelin sheath, your action potential can traverse quickly because it can jump, right? Whereas if you don't have a myelinated axon with neuron, for that matter, then it has a long route to travel. That's why you need myelin sheath. Because what is myelin sheath doing? Let's say this thing, action potential is coming here. It can't cross this myelin sheath. So what it will do, it will jump and it will go here. Again, it can't go here, so it will jump. So because it is jumping, it is reaching the end quickly. So it is a faster mode of propagation. Whereas if you see non-myelinated, there is no such sheath present. So it can simply keep moving one ahead the other. So it is relatively slower. Where it is present, how it is present, that I will discuss in the questions. Okay. And here you have the synaptic propagation. So you have, this is the synapse, this is the hollow space between these. So your action potential comes here. You have calcium 2 plus released into the neuron, and you have this neurotransmitter, which basically come and release. Now, how they release? So this is a neurotransmitter in basically a vesicle. So your vesicle will come and fuse with the membrane here, and it will basically open up. So your neurotransmitters move out, diffuse, and go to the postsynaptic. So this is called presynaptic because this is synapse. So this is the presynapse, and this is the postsynapse. Now, when this axon has to pass it to the next axon, this end will become pre-synaptic, and the next end will become postsynaptic. That's simply about it, right? Now, if any problem with these concepts, we will try to make sense with questions that I have put on the slides, so that you get more well versed with the topics. Okay, right. So, like I said, I will cover all the questions, all the topics that are part of the week. So let's begin with this, which is basically covering the first module. So just so you can revise. So the first one says which principle states all cells arise from pre-existing cells, and the second one says the phospholipid bilayer of the plasma membrane responsible for what? Why is it called? So there's a characteristic of the plasma membrane, and what is that? You can feel free to type in the chat box, because I mean, there's no judging, so you can feel free to type. Since we have like, oh, we are already half hour into the session, so I can take it like, I can wait for a few minutes and then we can move ahead in the session.
हेलो मैम, मुझे
Hello, yes.
जी इस मतलब एनिमल फिजियोलॉजी से संबंधित कुछ पूछना था मैम। मैंने क्या अभी ज्वाइन किया है,
Okay.
तो मुझे ये पूछना था मैम कि कुछ लेट तो नहीं हुआ, कुछ असाइनमेंट मेरे छूट गए।
So assignments are basically graded, तो उसमें मार्क्स लगते हैं. बट डू यू तो आप अभी जितने भी आपको असाइनमेंट मिलेंगे वो आप अटेम्प्ट जरूर सबमिट कीजिए ऑन टाइम. आपकी
और मैम
कितने
जो
आपकी
असाइन
आप कितने असाइनमेंट्स आपके नहीं हुए या आपके छूट गए?
मैम मैंने केवल आज ही से ज्वाइन किया, तो मैम वो तो आपको पता होगा कितने असाइनमेंट है, लेकिन मुझे अभी पता नहीं
राइट.
मैम।
नो, मुझे लगा आपने अभी रीजॉइन किया. सो आपको अवेलेबल हो जाएंगे आपके पेज पे. अगर नहीं तो आप इंस्ट्रक्टर को पर्सनली रीच आउट कर सकते हैं, आप मैसेज कर सकते हैं, वो आपको अवेलेबल कर देंगे. आपको बस लिखना पड़ेगा दैट यू हैव जस्ट जॉइन द कोर्स.
मैम थैंक यू मैम।
Okay, so let's me show you the answers. Let's just go ahead. And with each question, I will try to give you the background so that you basically connect it to the lecture this week. So it basically says which principle states that all cells arise from pre-existing cells, which is basically one of the topics that Professor discussed this week or in this lectures. That when you look at evolution of cells, all these cells have basically come from the previous cells. So the answer to these questions are basically this. So if you know about cell evolution, all cells come from a pre-existing cell. It is also called omnicellula acellula. It's basically one cell gives rise to the other cell. And because it is a biomolecule which is giving rise to another biomolecule, it is called biogenetic principle. And that is what it's saying here. Okay. Next one says the phospholipid bilayer of the plasma membrane is primarily responsible for selective permeability. Remember, the cell membrane is called a selectively permeable membrane. Why? Because we have phospholipids. So we have a hydrophobic and a hydrophilic head to the plasma membrane, and that does not allow everything to pass through it, right? So you have only certain molecules, certain ions that can move across the plasma membrane, and that's why it's called selectively permeable. That's a feature of the cell membrane, right? Okay, next one. So I will first ask you this question, and then I will explain what does excitable cell mean. So which of these following do you think is an example of excitable cell, and what is excitable cell means? For people who have joined the session first time, these questions are from previous assignment, previous exams, and some from previous workbooks and some new questions. So these questions are basically what can give you a guiding pathway when you are trying to revise. Okay, so all these questions are have been somehow part of the curriculum in this that way or this way. Right. So this is the first topic that we were discussing, excitable cells and non-excitable cells. So what do you mean by excitable cells? So the question asks here, which of the following is an example of excitable cell, and what is the main role of ion channels in excitable cells?
न्यूरॉन।
Okay. Yes. So what you can do is, there is two questions, so you can answer or you can type in the chat box also. I can look at the chat box, so I can also see from there. If you are comfortable with answering, I will take that answer as well. And I will wait for 1 or 2 minutes because we have other person also, and then we can discuss the answer. Okay. Okay, I can see someone typing. Let me just wait. Okay, fine. So I have response. So let me give you the answer and then I can tell you. Right. So the first thing is, what is excitable cell? Like I mentioned at the beginning, excitability is in terms of allowing the movement of ions. If the cell can allow movement of ion, it is basically able to get excited. And why are we even concerned about movements of ions? Because we are concerned with action potential that can only happen if your ions are moving across the plasma membrane. So cells which are capable of putting these in motion, or basically allowing the movement of ions, are the excitable cells, and are the very cells that can conduct action potential or propagate action potential, right? So out of these cells, because we have been so far reading, it's neuron, which is an excitable cell, and that's how it is able to electrically conduct the action potential. And when I say action potential, it basically is the movement of sodium and potassium ions. So your ion channels are responsible in getting those movements, and they allow selective ion to flow. Means a calcium channel or a potassium channel will only let a potassium ion move outside. A sodium channel will only let a sodium channel move across. So these are very specific channels. They only are respectively like they're only working for their respective ions. Okay. Okay, next question. So now, which of these following, this is a very easy question. Which of these following governs the resting potential in neuronal cells? So whenever we say minus 70 millivolts or minus 55 millivolts or whatever. So when we are talking about resting membrane potential, with which uh, ions are we talking about the potential with? And in a cell, which ion has the highest extracellular, like when you compare, which has higher extracellular concentration? So be careful when you are answering. Look at the names properly. Yes, I have answers. Nice. Okay. Anyone? There's one more answer. Do you want to attempt, or we can go ahead? Okay. A and C, exactly like you people mentioned. And of course, I'm sure you people already understood why it is sodium and potassium, because that's what we have been discussing. Why I told that you need to be careful is because of this. Okay. There is this N is nitrogen. So don't get confused. Be mindful of answer when you are answering. Sorry. Because these count for MCQs. If you get one wrong, it's basically your marks is gone. So be careful whenever you're answering. Okay, next. Okay, now we have come to neurons and important terminologies which can also make you understand the topic. So we have nodes of Ranvier. So I have already shown you what's nodes of Ranvier. So what does these mean? And then next question, which is this. Myelination in peripheral neurons is done by which cell? And this one, myelination in neuronal central nervous system or neurons of central nervous system is done by which cell? So this is PNS, this is CNS. You have myelination in both. Which cells are responsible for making the myelin sheath? We have done part of this question in the previous sessions also. And where are nodes of Ranvier present? BAC. Okay, I can see the other person typing. I will wait for your answer. Okay. Sorry, which answers? A and D. Okay, I have responses. Let me give you the responses. Yes. So it's B, A, and D. And if you got it wrong, it's okay. No worries. So like first one, I think everyone got it, right? Nodes of Ranvier are basically those central points where you don't have the myelin sheath, right? These two you need to remember. Myelination in PNS is, or peripheral nervous system, is by schwann cells, and myelination in CNS is by oligodendrocyte. So this is okay if you don't remember, just be mindful. These are something that you can revise. Okay. Okay. All right. So there are several names of the potentials that have been used here. Which one is correct? So the minimum stimulus, remember this term, the minimum stimulus needed to trigger an action potential is called what? P. Excellent. You guys are amazing. Right. So if you remember the potential, this is minus 55 MV. So your cell is resting at minus 70. When it reaches a threshold potential of minus 55, that's when you have an action potential, which is generated, right? Great. Okay. Next. So read it properly. The efflux of dash assists in dash membrane to resting membrane potential. Okay. Second question, depolarization of neuron is because of what? So we know depolarization. What is happening in these spaces? What ions are moving across inside all this? C and C and A and B. Interesting. C, C. Okay. I will walk you through it. Yeah, I will just walk you through it. It's okay even if you got it wrong. That's the whole idea of these sessions. Right. Efflux. And so in my previous session, I have told you, efflux is basically outside. This is in terms, if you would get, as you would study, hence in the coming days also, terms like afferent, efferent. Wherever you see the E, always remember E is for outside. It basically to moving outside. So here efflux, what is moving out of cell? Because we know sodium is coming inside cells. So what is moving out? Potassium is moving out. And when potassium moves out, you have repolarization, right? And when you have depolarization, that's the opposite. So so in case you get
look, sorry, कुछ नहीं.
Yeah, no problem. So again, just a cue for you if you can remember it like this. Depolarization is basically moving away. D means anything to move away from normal. So the first step has to be D and the next step has to be D. D means moving away from the normal state. Repolarization, or re, means to coming back to the normal state. First is to move out, and then first is to move away from the normal phase and then come back. So depolarization, and we need to remember depolarization is because of sodium ions. That's what you need to remember. Yeah, that's how you can remember it. So these questions help you grasp more of these topics. Okay. Right. So we know repolarization already. So just another question to help you understand that. So during repolarization phase, what happens? And when the inside of the cell becomes more negative than the resting potential, what does happen to the cell? So just imagine your ion concentration and then answer the questions.
Depolarization.
You can type also. It's okay. It's okay. You can type. No worries. Okay. The next one. Do you want to go for the next one also? Okay. Got one response. Yeah. C. See, that's what. If you think once it, so these topics, like I keep saying, they're very logical. So once you understand it, then all the questions become very easy. So that's like you got it right. So if you have repolarization basis, so like I was saying, just a second. Let me try to get that. Yeah. So action potential, this thing you need to remember. This is the most important thing to understand. So if you remember these curve, everything becomes visualized. You can visualize everything, right? So use repolarization, your potassium efflux is happening. And when the inside of the cell is more negative than the resting potential, which is basically this phase here, because this is resting potential, it is more negative here, it is called hyperpolarized. So you got that right as well. So great job. Okay. Right. Now we move on to the pumps. Okay. So first thing is, which ion pump help maintain the resting membrane potential? I discussed this briefly. And the second one is, which are the following rebalances? So basically, this question says, who among which channel of which pump helps in rebalance concentration of ion to resting membrane potential after inward sodium current and outwards potassium current? B and B. Okay. Anyone else wants to answer? All right. Okay. So B and D. So this is a pump. Okay. What is a pump? So what does a pump say? So I will break this term for you so that it becomes clearer to you. Okay. Sodium, potassium, ATPase pump. Sodium is a channel. Potassium is a channel. Why is ATP is a pump? What is pump doing in general? If you have a water pump in your house, what does it do? It basically takes water from the groundwater and put it up. Something it is doing with the use of energy. What is the energy in this sodium and potassium movement? It is via the use of ATP. So it is ATPase pump, right? So what the first question says, which ion pump help maintain the resting potential? Pump means anything which moves something, like pump is basically a gateway that helps move stuff in and out. So that is a sodium potassium pump. We take both sodium and potassium together because one alone does not do the activity. Just movement of sodium is not bringing it back to normal condition. So let's imagine if it was only sodium coming in, there will be no resting membrane potential coming back. If calcium is also not going out, if it's only the potassium that goes out, but there is no sodium intake, what's the point? It will not bring it back to normal. You need both sodium and potassium to go out and in, along with your ATPase activity. So that's why both the terms together, not just one single channel. Yes, the potassium channel opening is what starts the repolarization, but it does not bring it back. Both of these work together to bring it back to rest and because the repolarization can only happen after depolarization. So your sodium needs to come in so that potassium goes out. This is what it means. Okay. Right. And this brings to the number. How many sodium and how many potassium are being pushed out and put in? So sodium potassium pump maintains the resting membrane potential by pushing how many of each of these ions in and out? See, you guys are so smart. Right. So 3Na out and 2K in. Right? Why? Because when your polarization was happening, when your depolarization was happening, these guys were coming inside and these guys were going out. So the pump will do the opposite to maintain the resting potential. You have 3 sodium out and 2 calcium, so 2 potassium in. Okay, great. All right. Now this covers your model, the Hodgkin-Huxley model, right? So the first one says in an equivalent circuit, which is basically cell membrane equal to a circuit. So what does a cell membrane represent? Okay. And the next one says the plasma membrane of a cell can be compared to a capacitor because of which property? C and A. Okay. C. The next one. Let's take a guess. Okay. No worries. It's an absolute no. Just a second. Sorry. I know what's happening. Yes. Right. C and A. C, I think both of you got right. A is also, so if it is a capacitor, Y is a capacitor. Okay. So the first one said cell membrane is both capacitor and both resistor. Why? Capacitor is likewise said, it stores charge. How does this capacitor, like how does this membrane store charge? So see, imagine this is a membrane. This is an insulator. I told you it only allows selective material to pass through it, right? It is otherwise impermeable to other stuff, right? So it is basically insulating. But it has charge or charged ions both outside and inside. So it is at the it's at the covering of the layer, or it is at the end of the cell. And outside the cell also you have ions. Inside the cell also you have ions. Just in center you don't have. This is a characteristic of the dielectric layer, if you know of capacitor. So because capacitor is vertically storing charge, it is also storing charge because it has ions both sides, right? So that's how it's a capacitor. And it is also resistor because of the presence of ion channels in the cell membrane. So in the cell membrane, you have these ion channels, which basically allow the ion flow, that to selective ion flow. So because these ion channels can conduct, or they are resistors, you also have resistor in the cell membrane as well as the capacitor, right? That's how this is the logic behind it. Okay. Okay. Now, if you have to describe Hodgkin-Huxley model in one statement, which one of this would be the correct statement? Read the statement. Let me read it out for you. So, Hodgkin-Huxley model is a chemical model of neurotransmitter release. It's a mathematical model of ion conductance during action potential. It is a structural model of ion channel proteins, or it is a genetic model of neuron evolution. P. Exactly. Like I have described when I was talking about this model, it's basically a quantitative model of how does action potential move. So it's a mathematical model. Okay. All right. This is about propagation of the action potential now. Now, which are the following ensures unidirectional propagation of action potential? And the second one is, we have nodes of Ranvier, we know, which are in the non-myelinated zone. What are they essential for? Okay, I see A. Okay, I think it's one response. Do you want to give the second response also? Okay, so it's B and A. A as you already got. Now let me tell you what is this B. So the first answer is that what is the unidirectional propagation of action potential? We also discussed this in one of the previous session. So your action potential, once this generates, it has to reach the end. It cannot come back to the start. So end means towards the synaptic cleft. So the action potential has to go towards the synaptic cleft. It cannot come back. And what ensure that it doesn't come back? A refractory period of sodium channel. What is refractory period of sodium channel? So what does depolarization means? opening of sodium channels, and your sodium channels are sodium ions are moving inside. Now, once the sodium ions are moving inside, and I told you, right, they open, then they shut, then they close. So once they have put the Na plus inside, they shut. Once they close, they do not open again until that first bout of this action potential has propagated. So this is called refractory state, right? It becomes inactivated. So during this, they cannot reopen immediately, and that's why your action potential does not come backward. So it basically in a resting, it's basically, how do you say, retracting period. It doesn't open immediately again. It opens, push sodium and sodium channel ions, and then closes again, so that it doesn't open again for that another round of initiation. That's how you have only one direction of propagation. And nodes of Ranvier have saltatory, which is what I explained also. The action potential basically jumps when it is moving via myelinated axon. Okay, very interesting question. So velocity of conduction of action potential in muscles is dash times that of that in myelinated nerves. Please take your time, think upon the question. I will give you first few minutes. If you are not clear, you can try to read, and then you can try to attempt. It's a very interesting question. Okay, I have one response C. So, okay, I already have someone writing it. B. Okay. It's B, and I will tell you what goes behind it. It's very simple. Just that you need to remember this, okay? What does it say? It says velocity of conduction is basically how quickly your action potential is moving through muscles and in myelinated nerves. Muscles or skeletal muscles, they don't have your myelin sheath. So they have a slow conduction. Whereas your nerves, which are myelinated, they have extremely fast conduction. So myelinated nerves have 60 meter per second conduction rate, and skeletal muscles have 4.5 meter per second. So the question says muscle to myelinated, which is this skeletal to myelinated. Had it been myelinated to skeletal, it would have been the other way around. So let's first do the myelinated to skeletal. So myelinated is 60, skeletal is 4.5. 60 is to 4.5, or 60 divided by 4.5 is 13.33. If it is the opposite, it is 1 by 13. So that's the answer. So that's why I said it's very interesting. There are a few very important, what do you say, insights into this. You need to know the velocity or conduction rate of both the type of neurons that we have. Okay. So this hopefully is clear to you. Okay. Okay. Now we go to the another topic, which was extensively discussed in your lecture 5 or lecture of week 5, which is neurotransmitter. And in neurotransmitter, you had different kinds of neurotransmitter: excitatory neurotransmitter, inhibitory neurotransmitter. So we have all of these put up in different questions. And with each question, I hope it becomes more clearer to you. So first of all, let's begin with what is a neurotransmitter. Which one of these is not a neurotransmitter? This is the easiest question. It's laminin. Okay. And when you see the neurotransmitters, glutamate and GABA are very, very important players. Glutamate is basically a major excitatory neurotransmitter for the CNS. GABA, which is this, is an inhibitory neurotransmitter. So this you need to remember, excitatory and inhibitory. And this dopamine is actually a neurotransmitter, which is how do I say? It's in we have studied this in motion, in movements. So dopamine is also a neurotransmitter. Now, what is excitatory and inhibitory? It's basically telling the how the action potential should propagate. If it's excitatory, it is taking you to depolarization state. If it is inhibitory, it's inhibiting it. That's it. That's what the name suggests. And why is it important? Because when your synaptic cleft has these neurotransmitters released, these neurotransmitter will tell the next neuron what to do. Should you get excited or should you inhibit the action potential? That's the role of it. Okay. While what is this lamin? This laminin is actually a protein. It's a glycoprotein. So you will see this in the extracellular matrix, ECM. Okay. All right. So I told you about one inhibitory neurotransmitter. Could you guess from here which is the correct option? Great, excellent. Yes. C, GABA, right? So your acetylcholine, the other name that we are seeing right now, acetylcholine, it's a excitatory neurotransmitter. Remember this. And I think if you had read about synaptic cleft and if you have synapse transmission, acetylcholine is something that you definitely know. You know there are acetylcholine receptors on the cells, right? So acetylcholine is an excitatory neurotransmitter, but where is it present? It's present in the muscular. So it's a neuromuscular junction transmitter, right? It's a name that we have read a lot of time. Glutamate, as I have just told you, is an excitatory neurotransmitter in CNS. Hopefully, you're able to locate where these neurotransmitters are acting. Acetylcholine is acting in the neuromuscular junction. Neuro acting with the muscles. Glutamate is working in the CNS, in the central nervous system. Okay. GABA is also working in the central nervous system but as an inhibitory neurotransmitter. Dopamine, on the other hand, is a modulatory neurotransmitter, right? It modulates our behavior. Okay. Okay. Now, CNS done. I've spoken to you about neuromuscular junction. What about brain? Any idea about brain, which is present in brain? Okay. Do you also want to attempt? Okay. Let me show you. Just a second. Okay, let me show you the answer. It's glutamate again, and that's why I asked, like, so that I could bring this in this thing. So glutamate is present in brain as well. Glycine, the two names that we are seeing for the first time, glycine and serotonin. Glycine is actually inhibitory uh, neurotransmitter in brain. So in brain, glycine is the inhibitory neurotransmitter, and serotonin is again a modulatory neurotransmitter. Now, I hope here you get the word modulatory is clear to understand. Do you know what serotonin does? It helps your elevate your mood. It's a good thing. It helps your sleep regulation, your appetite regulation. So these neurotransmitters modulate what kind of physiological responses we have. That's why they are modulated neurotransmitter, both dopamine and serotonin, right? They are not part of this relay of information in this CNS of major excitatory for the action potentials. Okay. Okay. Now, when I say inhibitory, that's what is more important, right? When I say inhibitory, what does inhibitory mean? Any idea? It's a very interesting mechanism. And D, okay. Do you also want to take a guess? Okay. So let me give you the answer. It's chloride influx. And I will tell you how beautifully it connects. Okay. So your GABA is a neurotransmitter. GABA, all this while I've been talking about GABA. GABA, what is the full form of gamma? It's gamma aminobutyric acid. It's a component. Okay. So it's a neurotransmitter. It is released into the synapse. And as I told you, presynaptic terminal, release of neurotransmitter, postsynaptic terminal, you have receptors, you're binding it to the neurotransmitter. So your GABA is now released, and your GABA has receptors on the postsynaptic terminal. They are they're interacting with the ligands. So GABA and GABA receptors, they work. So there are two types of receptor. Let's just talk about one. So GABA A receptor. These GABA A receptors are chloride channels. Chloride, if you know, is written as Cl minus. Okay. So as soon as your GABA binds to the receptor, these channels open up. So they are binding, the GABA is binding here, and these channels open up. Whenever these channels open up, there is a, what do we read? There is an influx. So your chloride is coming in. Chloride carries a negative charge. And as soon as your negative charge is coming inside the cell, your membrane potential is decreasing. And your membrane is now becoming more negative. So you cannot, so see, your resting potential was minus 70. More addition of negative ions means is going below minus 80, minus 90. For from there, for it to do a depolarization or generate an action potential is very difficult. That's why it's inhibitory. That's it. That's very logical to understand. You have a negative ion channel opening, and that's how it's entering. Calcium 2 plus, I think the answer, the second answer that I saw was calcium 2 plus. Calcium 2 plus has a major role in the neurotransmitter excitation or exocytosis, not here. Here you need to inhibit the transmission of action potential. So you need to make the cell more negative, or you need to make it somehow possible that action potential is not generated. And that's only possible if your negative charge goes way below. That's the logic there. Okay. All right. Now, the next type of question, one we have done neurotransmitter. The next topic was neurons. The type of neurons is also, I mean, the type of neuron is basically on the type of chemical they're or neurotransmitter they're associated with, right? That's how they are named. So here you have the question. So which type of neuron will release acetylcholine at the neuromuscular junction? So your neurons, which come to a neuromuscular junction, which transmitter it release if it's, and which, so if it's called, if it's releasing acetylcholine, what it will be called? Easy, no? It's in the name itself. See the name before nergic. The term before nergic tells you what type of neurotransmitter is being released by the neurons. One more minute. Okay, let me give you. So acetylcholine is choline, so cholinergic. GABAergic means GABA releasing neuron. Dopaminergic is dopamine, and serotonin means serotonin releasing neuron. Okay. Okay. Now, if I say dopaminergic neurons, which is basically which is secreting neurodopamine, then what is dopamine associated? This is where your role of dopamine will come, and what I said about modulatory transmission will come. So, can you guess which one will be the correct answer here? Okay. Anyone else? Okay, maybe I will give you the answer here. So it's motor control and reward pathways. So your dopamine is responsible in these modulatory activities. Okay. Okay. Very interesting question. So there are pair of neurotransmitter here. Which one is an excitatory to inhibitory? So read the word and then say which one is the correct answer. So the combination is excitatory to an inhibitory. Which combination fits here? Fits here? Okay. I've got one response. Other? So let me show you the answer maybe. Okay, give you the answer. So it's glutamine and GABA, as you correctly mentioned. Now, why not the other option? So acetylcholine is excitatory, glycine is inhibitory, but the pairing is not correct. Whereas acetylcholine did we discuss neuromuscular junction, whereas glycine we discussed spinal cord, right? So they are not the same target, or they are not acting at the same place. Serotonin and dopamine are modulatory neurotransmitters. Okay, they can modulate behave based on the context. And GABA and glutamate are the opposite. So that's why glutamate and GABA. So you got it right. Okay. Now this is about the last topic, which is transmission at the synaptic junction. So what is the gap between presynaptic and postsynaptic called? And here you have calcium influx, which is basically doing what? Okay. Anyone else? Right. B and B. B and B. That's fantastic. Great job. Right. So your junction between or gap between the two junctions is called synaptic cleft. And here you have vesicle exocytosis. So vesicle fuses, the neurotransmitter is released. Fantastic. And next and the last question probably for the last topic here. We have more questions, which is, as we know, assertion reason based. So the spread of action potential in a neuronal network is mainly influenced by what? So when you tell your action potential is moving, propagating, what adds up to that propagation? What is responsible for that propagation? How will you bring it in one line? Or spread, rather. Propagation and spread both. B. Okay. B. Fantastic. Guys are very smart. Right. So you have both excitatory and inhibitory inputs. So both of them will together be responsible for the overall spread of action potential, right? Great. Okay. Now let's move to some different kind of questions. Right. People who have been in the sessions first, they know what questions are these. People who have joined for the first time, these are basically questions which are two statements. First statement is a fact, for example. Now, the second statement, does it justify the first statement, is what you need to answer. Okay, those are four options. So, for example, the first statement here is excitatory neurotransmitter depolarize the first synaptic membrane. And the reason for this is they increase sodium influx, which makes the cell less negative. So, read the two statements and tell which inference is correct. Think, what is depolarization? What is happening? Is that why you are calling them? Uh, that's what is relating the two statements. Okay. I've received one response. Do you also want to respond? It's A. I thought C, right? R is false. Okay, no problem. So first thing is true, we all agree. Excitatory means that it is able to generate action potential. So it will depolarize the postsynaptic membrane, which is basically the next neuron. Okay. And why are they depolarizing? Why are they depolarizing? Because you have sodium which is coming inside the cell. The sodium channels open up where there is a sodium influx. And sodium influx means more of potassium inside the cell, which makes it less negative. So your membrane potential increases to plus 40. So that's why these both are correctly explaining each other. Okay. All right. Next one. GABA reduces the probability of action potential generation. It causes hyperpolarization by allowing chloride influx. We have just discussed this. This is to put in a more formal way. So, which of these answers is correct? We just discussed, right? Okay. Let me. Okay. I see one response. Do you also want to attempt? Yes. Correct. See, you people remember stuff also. Right. So you have two statements, GABA, which reduces the probability of action potential generation, and it causes hyperpolarization, which is basically like, I remember we told that GABA binds to the GABA receptor, opens the chloride channel. Chloride comes inside, more of negative charges inside cell. It goes way negative in its membrane potential. It becomes hyper negative, which is this hyperpolarization. And it is not able to generate the action potential anymore, and that's how it inhibits the action potential. Fantastic. Okay, next one. Calcium ions are essential for neurotransmitter release at synapses. Calcium entry activates vesicle docking and exocytosis as the presynaptic terminal. Okay. One response. Hmm. Okay. It's correct, right? So we all agree that both the statements are correct. Now I will tell you why is our or second statement explaining the correct first statement. So calcium ions are essential for neurotransmitter release. And how do release happen? So neurotransmitters, as we know, they are bound in these small vesicles. For neurotransmitter to be released outside, the vesicles need to pop up. That popping or that, how do I say, breaking of the vesicle takes place because of calcium. So that's why calcium is responsible for neurotransmitter release. Because when there is calcium entry, or when your calcium enters your neurons, your vesicles, they dock and bind to the membrane, and there is exocytosis. So the neurotransmitter is released. This neurotransmitter is released because of calcium via vesicle docking. So that's why this is the correct statement. Okay. Right. Next, serotonergic neurons only have excitatory effects. Serotonin always depolarizes the postsynaptic neuron. What do you think? Part of this we had just discussed in the previous mix and match question. I mean the pair question. So if you can recollect. Okay. C. Okay. Other? Okay. C. And anyone else? Okay, no worries. I will give you the answer, which is this. Okay. Yes, it's confusing. So that's why I kept it here. So it says serotoninergic neurons only have excitatory effects and they always depolarize. No, we remember what is the name for these transmitters? They are called modulatory neurotransmitters. So they modulate and they can have both excitatory and inhibitory. So they can both depolarize and not depolarize. So they can do both. So they're not restricted to only one type. That's why both the statements are incorrect. Okay. All right. Next. Okay. Sodium potassium ATPase pump maintains the resting membrane potential. It actively pumps 2 Na plus inside and 3 plus outside. Which one do you think is correct? Oh, which one do you think explains it? Okay, C. Anyone else? You want to take a guess? Okay, no worries. Let me give you the answer to this. Okay, so yes, false. First statement is correct. Yes. Second statement, read it very carefully. It says it pumps 2 Na plus inside and 3 K plus outside. Does it require pumping for that? No, right? Pump uses and pushes in the other direction. It pumps 3 Na plus outside and 2 Na plus 2 K plus inside. Okay. Yeah. Next. The phospholipid bilayer or phosphobilipid layer of the cell membrane allows free movement of ions across it, and the hydrophobic core of the bilayer prevents diffusion of charged molecules. Read very, very carefully which statement says what. D and D. Great. Exactly. And why D? Why is it false? Because it does not allow free movement. It's a selectively permeable membrane. Fantastic. Okay, next one. Resting membrane potential of the most mammalian cells is around minus 70 millivolt. It is primarily from the selective permeability of the membrane to K plus ions. Anyone? B and A. Okay. It's A. Okay. And which basically is that it is because of the K plus permeability. Okay. Okay, next one. Excitable cells include neurons and muscle fibers. They can rapidly change their membrane potential in response to stimulus. Which one do you think? Okay, I have one response. Next response. Okay, very good. Right, that explains, right? Because they are excitable because they can change their membrane potential. Okay, next one. The resting membrane potential is usually negative. It is due to high intracellular Na plus and low extracellular K plus. Think. Hmm. Okay. Anyone else? Just to response? Okay. Well, that's C. And as you got it right, see, it says the resting membrane potential is negative. Yes. It's high intracellular Na plus. No, right? Remember, actively you are pushing out Na plus. So intracellular is incorrect. You have high extracellular Na plus and low or high intracellular. So this is basically reversed. Why? Simple. If you want to understand, just remember what is happening when there is depolarization. Depolarization is basically diffusion. Okay, your ions are diffusing. If your sodium ion is when the channel is opening, it is moving inside. Eddy diffusion is from high concentration to low concentration. So if your depolarization, sodium ions are moving inside channel, it means sodium ions are very higher outside, and that's why it is coming inside. Similarly, when your potassium ions open, potassium ions move outside because they're high inside and they're low outside, right? But when you are doing using energy, it's basically you're doing something opposite. So when your ATP pump comes into play, what it does, it just reverses the entire thing. Whatever came inside went outside. Now, when sodium is coming inside, it doesn't mean that sodium ions are becoming equal or they are opposite, changing in the opposite direction, right? So what it does, it is still less inside. So for it will not go by diffusion outside. So the sodium pump has to push this sodium, which is inside to outside. So it is against the diffusion process. So that's a unit energy. But before the pump comes into play, it is following a diffusion process. So that's why just remember the first directionality, and that will tell you which one is correct and which one is incorrect. That's how you remember it. Okay. Next one, depolarization occurs when the membrane potential shifts towards positive values. This happens because K plus enters the cell. These are all easy questions, but yes. Do you have answers? Oh, sorry. Okay. So I've got C and C. C, that's fantastic, right? Why? Because we know what is coming inside the cell, Na plus is coming inside the cell, right? By now, we have done so many questions that I am sure you would have got very well versed with this topic now. Next, hyperpolarization makes a neuron less likely to fire an action potential. It increases the distance from threshold potential. Very easy question. Okay, anyone else? Okay, I think you will not receive response. Think upon it and what do you think is the correct answer? Okay, I've got received one response. Other? Great. Okay. Yes, we know that, right? Because you are now more negative. You are at minus 90. So from going from minus 90 to plus 40 is very difficult. That's why you're less likely to fire an action potential. You inhibit it. Okay. Next, the sodium potassium pump consumes ATP. Movement of Na plus K plus is an energy independent passive process. Pump, okay? We are talking about pump here. Let me give you the answer for this. Sorry. Where did it go? It is C. Okay. What responses did I see? Wait. B and C. Okay. So A is true, right? And when it is consuming energy, I just told you if it is consuming ATP, ATP is energy currency. So it is ATP dependent process. It's the energy dependent process. That's how what I meant. Okay. And if it is energy dependent, it is not passive process. If it is consuming ATP, when it is doing the pumping, it is basically using up energy. Okay. All right. Myelination increases the speed of action potential propagation. Myelin allows saltatory conduction, where the action potential jumps between rods of Ranvier. These are all getting easier now because we have done enough questions to understand it better. Yes, exactly. A, right? Because that's what we have discussed. Because of the nodes of Ranvier, it can jump quickly. Next, action potential travels faster in myelinated neurons than unmyelinated neurons. Myelin sheath allows solidity. So it's basically the same kind of question. Just so I often keep giving you same kind of question, not so that I repeat the topic. It's just basically making you aware of all the different kind of words that can be used for single topic, so that you whenever you read those topics in all forms, you are able to understand the question. So all these are the same kind of question for one single topic. They look repetitive, but that's the whole agenda here, that I try to cover as much as I can for the topic. Yeah. Okay. One. Uh, right. So A, right? We have done this already. So the difference between the first and the two one was just the uses of words interchangeably. Okay. Next, neurotransmitters are stored in vesicles in the presynaptic terminal. Vesicles prevent neurotransmitter degradation before release. Okay. Yes, precisely, right? Because that's how your neurotransmitter are present in this vesicle. Because with the vesicle protects it from all the lysozymes and the enzymes which are present in the cell. So if it's not present in this vesicle, in this covering, then those will degrade. It will come. They will attack the structure, or the enzymes will break them down. That's why to protect them, they are present in vesicles. Okay. Okay, GABA and glycine are inhibitory neurotransmitter. They increase chloride ion influx, causing hyperpolarization of postsynaptic neurons. Yeah, great. Okay, we know this already. Okay, now at a chemical synapse, neurotransmitter depends on calcium influx at the presynaptic terminal. These calcium ions bind to synaptotagmin, triggering vesicle fusion with the presynaptic membrane. So the mechanism is what we were missing. So that question tries to question, this question is for that part. Okay. Anyone else wants to answer? Okay. Yeah. Let me give you the answer, which is correct. So remember this. I just told you that when calcium is there, your vesicle is doing exocytosis. And I have also told you before that the vesicle first comes and fuses with the membrane. That fusion is because of the synaptotagmin. Okay. Okay. Now, so, okay, I will first ask this question and then I will explain. So electrical synapses allow faster transmission than chemical synapse, and they rely on gap junction that permit direct flow of ions and small molecules between neurons. So basically, this is what I wanted to say, that there are two types of synapses, electrical and chemical. So we'll discuss that. Just answer it and then I will discuss this. Okay. Sorry. Yes, right. So you have electric synapses and they are gap junction. So what is electric synapse and what is chemical synapse is basically, so the electrical synapse are when the neurons are connected by their cytoplasmic connections. And because it is cytoplasmic, it is traveling faster, right? Because there is direct flow of ions between the neurons. Whereas in chemical, they have to go through that synaptic cleft and then they have to go via the neurotransmission, right? So the neurotransmitters are the chemicals, that's chemical synapse. And because it has to go via the cleft, there is this distance between the two neurons end and start. That's why they are slower. But in electric synapses, they are collected by the cytoplasmic interconnections. And that's why via the gap junctions, quickly the ions can move from one to other and propagate the action potential. And that's why they are faster because they don't have to rely on any distance, right? Okay. Okay, myelinated axons conduct action potential faster than unmyelinated. Myelin sheath increases membrane capacitance, allowing faster conduction. Which one do you think is the answer here? Okay, C. Any else answer? Okay, C. Okay, let me give you the answer. This is a very smart crowd. Exactly, C. Because myelin sheath does not increase membrane capacitance, it decreases the capacitance. And because it decreases the capacitance, it increases the resistance. And because resistance means conduction, so you have more conduction of the ions. Great, fantastic. Okay, during an action potential, depolarization occurs due to opening of voltage-gated sodium channel. Sodium ions move into the cell because the equilibrium potential of sodium is more negative than the resting potential. Just try to imagine in terms of the concentrations or the flow of iron. Okay. Okay. That's it. Wait, let me give you the answer. Like someone said, it is C because your equilibrium potential is more positive than negative. It's not negative, it's positive. Okay. So, and that's how you have ions moving inside. Okay. All right, I think we are nearing the end. Yeah, few more questions. So, potassium efflux during the falling phase is lower compared to sodium influx during depolarization. Voltage-gated potassium ions activate more slowly than sodium channels. Yes. Read the two terms and then think. We already discussed this. Do we like this is already unknown to us? Just read the terms very carefully. Hmm. C. Okay. Any guesses? Okay, the answer is A. Okay, right. So potassium efflux during the falling phase is lowered compared to that. We know this already because that's how you have a hyperpolarization, right? There are more which are negative, which are moving out. So during a normal phase, sodium is coming in, potassium is going out. Your potassium going out should close after a while, but we know that the closing of potassium channel is slower. That's why you keep having more potassium outside. But this also means that the voltage gated potassium channel will activate more slowly, because voltage gated means the ones that get activated to do the opposite so that they can begin to resting potential. What happens after hyperpolarization? That to come to resting potential, right? So that coming back, or that stopping of the movement of going of potassium outside, will be stopped by the voltage gated channel. They also activate slowly. So both the things are happening at a slow scale. That's why both together lead to that large drop in the potential, which brings it to hyperpolarized level. Okay, so voltage gated channels are opening, activating slowly, and the origin and the channels which are responsible for bringing or moving the potassium outside, that are also closing slowly. Okay, right. Okay, now multipolar neurons. I will, you first answer and then I will explain the term. Multipolar neurons are the least common type of neuron found in CNS, and they have one axon and two or more dendrites, allowing integration of large amount of synaptic input. Now, yeah, the term is given to you, and you know how important CNS is. Do you think these make sense? C and D. Okay, wait, the answer is D. Okay, the opposite. This multipolar neurons are basically, as the name suggests, multipolar. So neurons have many dendrites in the end. And CNS, central nervous system, is a region where you need great amount of synaptic input. So that's the whole reason you have all those dendrites, so that you have those more how do I say, more interaction between different neurons. So multipolar neurons are actually the most common type of neuron in CNS. And the second reason is because they have this one axon and multiple dendrite that's needed for great amount of synaptic inputs. So, and you need that in CNS, right? Okay. I think this is the second last question, and then two more questions and we're finished. So the equilibrium potential for potassium is more negative than the resting membrane of the typical neuron. At rest, there is a small inward sodium leak that depolarizes the membrane. Hmm. We discussed about sodium, right? We talked about equilibrium potential being positive or negative, whichever. If you remember that discussion, this can help you answer this question. Think. I think, yeah. Two more questions. Yeah. A. Do you also want to attempt? Fantastic. People are excellent. Yes, it's A, right? We already discussed the other section that sodium equilibrium potential is more positive. That's why it comes inside. And that's how equilibrium potential for potassium is more negative. That's why it moves out. And to make it more negative, you have this leaky channel that pushes some sodium inside, making it more amenable for it to move outside, right? Fantastic. Okay, the second last question for the day. So the Hodgkin-Huxley model is not sufficient to explain action potential initiation at the axon hillock in mammalian cortical neurons. The model does not account for high density sodium channel clustering and other channel types such as calcium potassium channel. Now, this looks very heavily worded, but there is just basic two things here. If you remember the lecture notes also, this was mentioned there. So I just want you to answer it first and then I can tell you. Okay, let me give you the answer for this. So this is A. Okay. So what does it say? That this model is not sufficient to explain action potential, right? So if you remember in the lecture notes, it was said that this model, Hodgkin-Huxley model, is a quantitative model, right? It explains how it is initiated and how it moves, but it does not explain the mechanisms behind it, right? And it does not do that because when this model was proposed, we did not know about the gated channels and all these ion channels. So this model only tells you, yes, it is propagated and this is the rate of the propagation, but how it is propagated or how it is generated, we did not know, right? And this is what it is saying, that it is enough to tell you, yes, it is propagated, how it is moving, but mechanisms is explained by other channels and other features which are not part of Hodgkin-Huxley. So just looking at the model will not explain the action potential in its fullest. You need this channel and pump information to explain it in a whole way. Okay. Yes. And that brings to the last question, which is taking from here. So the Hodgkin-Huxley equations are deterministic and predict identical action potential for identical stimuli, right? The model does not incorporate stochastic channel gating, which can cause variability in real neurons. What do you think is the answer? This is the last question for the day. Yes. Do you also want to attempt? Fantastic. Yes, that's exactly what it is. Because it did not know about gates, because it did not know about pumps, it said that if the initial stimulus is same, the rate of action potential's propagation will be the same. But that's not how it works, because you have channels, you have leaky channels, you have some different channels and different voltage tests which give rise to different kind of propagation rates, right? Because it was not incorporated, the model says if the stimulus is same, everything else remains same, right? Yes, and that brings us to an end to this session. If you have any question about any topics, you can post, or else we meet each other in the next session, which is next Tuesday, no, next Monday. Sorry. Thank you. Yes.
हेलो मैम,
Yes, yes.
मैं अपने इंस्ट्रक्टर से कैसे बात कर सकता हूँ मैम?
हमारा ना फोरम है, तो आप फोरम में अपना क्वेश्चन पुट कर सकते हैं एंड द इंस्ट्रक्टर विल रिप्लाई टू यू.
मैम मैं समझा नहीं, ये फोरम में मतलब ईमेल आईडी पे?
हाँ, so in your NPTEL, when you open the interface, you will have a place to post your question. अगर आपको नहीं मिलता है, just let me know, I will help you. आप so this लिंक से आप इस चैट बॉक्स में आ सकते हैं और आप कब मुझे मैसेज कर सकते हैं. I can show you how to do it. Just try. आपके इंटरफेस में आप जाएंगे तो वहां पे ऐसे Q&A लिख के आता है. You can go there and you can type your question. It will take you, it basically takes you to the forum, जहाँ पे लोग बहुत सारे डाउट्स पूछते हैं. So you can ask your question there. आप ट्राई कीजिए, नहीं होता तो आप मैसेज कीजिए. I can help you with that.
ओके मैम, थैंक यू मैम.
Thank you so much. Thank you.
थैंक यू मैम, थैंक यू.
Thank you.